In the year 1917, Albert Einstein first described the theory of stimulated emission. Little did he know that this work on atoms and energy laid the foundations for a multi-billion-dollar industry. We're talking about lasers. Lasers release light, but they're not the same as light bulbs or flashlights because they produce very narrow beams of light. If you would grab a flashlight right now and point it towards the wall, you would notice how some of the light gets lost on the way to the actual wall. It's as if it disperses itself. If you would grab a laser instead and point it to the same wall, you would see a small, shiny dot on the wall, and your cat would probably chase it. When making lasers, scientists use light waves that have very similar wavelengths. The light waves are coherent, meaning the beam of photons moves in the same direction at the same wavelength. This is why laser beams are very narrow, very bright, and can be focused onto a very tiny spot, just like in our imaginary wall. We use lasers to do many activities. Doctors use them in very precise surgeries, workers use them to cut minerals like diamonds, and physicists can even use laser technology to record and retrieve information, even information at the microscopic level. Hello and welcome to another episode of Crosstalks. My name is Barbara Pinho and I will be your host. This is a podcast series by the UT Austin Portugal Program that will walk you through the wonders of science-business collaboration. We will be exploring emerging topics in advanced computing, medical physics, nanotechnologies, space-Earth interactions, and technology innovation and entrepreneurship as we delve into the program's industry-driven research projects. In today's episode, we're talking about laser technology that helps us see what our eyes cannot. Laser microscopy is a technique used for generating microscopic images by scanning objects with a laser. Light carries information from the sample, and with this data, a computer produces images that showcase the microscopic objects. These objects can either be cells, living tissues, you name it. Now, there are some limitations to what we can see even with the most advanced technologies. Scientists can already see some cellular mechanisms deep in tissues. However, some interactions between molecules, such as protein-protein interactions, are still hard to catch. Within the ExtreMED project, scientists are developing a more powerful technique. They aim to have a fully operational cycle laser prototype by 2023. Curious? Let's hear the experts. They are Rosa Romero and Andrew Dunn. Rosa Romero is the CEO of Sphere Ultrafast Photonics and has extensive experience in technology transfer. Her expertise in lasers started 17 years ago, working in non-linear propagation and laser-matter interaction. Currently, she is devoted to making new solutions for the ultrafast laser market. Andrew Dunn is a professor of biomedical engineering at UT Austin and the director of the Center for Emerging Imaging Technologies. His research focuses on developing novel optical imaging techniques for imaging brain functions. So welcome, Rosa and Andrew, and thank you for accepting our invitation. How are you?
I'm good.
Very well, thank you.
Well, I hope you're excited to be with me on this podcast today. So to kick things off, I know both of you have been working with lasers for years now. When was the first time you actually got interested in working with lasers? Rosa, can we start with you?
I was an undergraduate and I just got fascinated by the light and the colors of the laser systems on the research lab that we visit on our second year. And since then, I follow the photonics, let's say, path.
Where were you studying?
In Santiago de Compostela, in Spain.
And what were you studying, undergraduate?
Physics.
But physics has lots of exciting stuff, doesn't it? Space and all of that juice. What made laser special? Was it the colors?
I think it was everything. It was like you could be able to see that the light, that the speckle of the light. I got fascinated by it. So I wanted to know more about it and know how to use it to do things. So that was making me excited.
Lovely. What about you, Andrew?
So I think my very first exposure to lasers was when I was a child. I heard about my father having laser surgery on his eyes, and that sort of fascinated me. Of course, I knew nothing about lasers and how they worked, and hadn't actually ever seen one, but I heard about them, and it just seemed sort of amazing to me that they could be used to treat somebody. And then later on, my first actual exposure to lasers was similar to Rosa's, and that is as an undergraduate physics major. And it sort of just grabbed my attention that they could be used for so many different applications.
Yeah, indeed. And perhaps listening to someone you care about having a surgery with something that seems so shiny and perhaps not that much understandable by a kid, right? It's quite interesting. And I understand that the project you both are working on with other people, ExtreMED, I am hoping I'm pronouncing that properly. I understand that you're using lasers and you're going to make more stuff with lasers. Now, for people who are not that familiar with physics, please bear with us and let's try to walk through the delicacies of this project. And I'd like to start with asking you, Andrew, could you please just explain to me briefly, what is this project about? And Rosa, if you have anything else to add, go for it.
Yeah, so it's a really kind of exciting and fun collaboration between some academic research labs and company, where we're trying to use a new sort of the next generation of ultrafast lasers for a variety of applications, most of which are centered on biomedical applications. So using these lasers to see aspects of biological function that we couldn't see before. So really as tools to advance our understanding of how the body works.
Rosa, would you like to add to that?
Something that people could understand is that with these new kind of lasers, very, very short lasers, sub 10 femtoseconds, so it's really, really short. For example, you damage less your samples, you can image deeper, and this is because of the nature of this laser. And you can also, for example, scan faster a sample. So I would say that these are adjectives that people could understand very well and see the advantages of using these, let's say, new generation of laser systems.
That sounds quite interesting. And I know Andrew mentioned that there's this fun cooperation between academia and a company. I understand, Rosa, you're here for the company, right? Can you talk a bit about what is your company about and how will it lead this project towards a new prototype?
Okay, so the company Sphere Ultrafast Photonics is focused on pulse-temporal diagnostics, but we are a company that was born in a group that design and make laser systems. So this was the struggling for the group to be able to measure the temporal duration of the very short pulses that we were designing at the university. And all these projects start about because we want to find an application, a mass market application for this generation of very, very, very short pulses. And well, we know very well INL. We work very closely with University of Porto. We have the opportunity to meet Adela some years ago at Austin because we knew she was doing some scalpels with femtosecond laser systems. And then Adela knew Andy. And so we make this consortium. So I really I'm really glad that we were able to make it.
Yeah, I see. So Andrew, how about being the person in academia within this partnership? How does it feel?
It's, as I mentioned, it's a lot of fun. Being able to work with a company like Sphere is great for us because it allows us to gain access to really cutting edge new products that can ultimately help our research.
Do you feel you can be more creative with the work that you want to do because you have this partnership?
Absolutely. And in fact, part of that is the result of these ultra-fast lasers really open up all kinds of new possibilities of types of research questions that we can ask and hopefully answer. So absolutely, I feel like we can be more creative. And in some cases, sometimes it feels like we have to sort of limit ourselves because there's so many possibilities, but that's a good problem to have.
Oh, that's a good way to summarize it. It's a good problem to have. You did mention multi-photon microscopy. So what is this? How does this type of microscopy work?
So it's a type of microscopy technique that produces three-dimensional images of biological samples. And one of the key aspects of multiphoton fluorescence microscopy is that it requires ultrashort laser pulses. And the term ultrashort means something fairly specific. It means laser pulses that are on the order of 10 to the minus 15 seconds in duration. So those are very, very short pulses, so femtosecond pulses. When you confine light in time to such a short period, the physics, the resulting physics restricts the generation of fluorescence to a very, very localized spot in space. So if you then move that spot around your sample, you can produce three-dimensional images of fluorescence images of samples. And this is used to image in vivo, so living tissues, as well as in cells.
That sounds quite interesting, but I would like to understand what problem would this actually solve? So Rosa, you did mention that this would let us see more and research more and see samples with new pairs of eyes, but what practicality comes out of this?
So you don't kill your samples. So as a scientist, you spend a lot of time preparing the samples, putting on the glass, and to put it on the microscope, or even if you have live cell samples, you don't want to kill them. And the problem nowadays is that with the type of femtosecond lasers that you are using with longer temporal durations, they apply more heat to the sample and they end up by dying faster. And one advantage of having shorter pulse durations is that you don't heat the samples so much, and you can scan it multiple times, for example.
Is it the thing you mentioned about not killing the samples because there's not as much temperature? Is that related to the fact that we have short pulses, as both of you explained? Okay.
That is correct.
That does make sense. So this would be very practical for scientists, I'm guessing, and researchers using this laser to look into samples. But is there any other applicability?
Yes. So for example, you can develop new techniques. For example, with the shorter the pulse, the wider the number of frequencies, the number of colors that you have on your source. And for example, now in microscopy, multiphoton microscopy, they use very narrow, let's say, bandwidths to excite different parts of the cell. So they have different excitation bands. And with a shorter pulse, you can excite all these bands, for example, at the same time, which also implies that the sample lives longer because you don't have to scan the sample multiple times with different type of lasers. You can use only one laser source instead of having 3 or 4, depending on the amount of fluorophores that you want to excite on your sample.
Mm-hmm. I am seeing this as a very useful tool for researchers in the biosciences. But would this actually be relevant for treatments?
Yes, in principle, yes. But Andy, go ahead.
Yeah, so it is possible. As you mentioned, the primary use of these lasers in microscopes is, I would say, on the preclinical research side. And in fact, they're very, very widely used now as tools for studying basic biological processes. So it really in research. The clinical applications are lagging the research applications so far. But there's a couple of clinical applications that hopefully will become widely used, and those are, one is for actually treating tissue. So you can actually sort of use some of these ultrafast lasers to very precisely cut tissue, but you can also use them in ways that are similar to the research applications and potentially image tissue as well with very, very high resolution. The challenge there clinically for the imaging applications is that in general, you need fluorophores, so the fluorescent agents, contrast agents that provide a source of signal. And at least in the preclinical models, those come from dyes, essentially, that you introduce into the tissue. That's very, very difficult to do in humans.
Clinically
because
speaking,
there are
right?
very
Getting
few
the dyes there. Oh,
approved for use in humans. Hopefully that will change in the future.
I understand. So this would be a feasible treatment or something, a tool to be used in treatments if we get the dyes and ways to actually signalize what we're trying to look at. But I understand you're actually building this tool and you're going to make a new prototype. It's one of the goals of this project. And I was wondering, where will this be built? Will it be here in Portugal? Will it be in Texas? Where are you planning on doing this?
So we are building several prototypes to be able to apply this technology, these laser systems to different applications, as Andy was mentioned at the beginning of this session. So basically we have here a laser system that is going to travel to the US, for example. So Andrew and Adela can do tests with these lasers on their own applications, for example, in fast scanning or non-degenerative microscopy imaging. So it's a different type of microscopy. So the idea is to explore where we can go with these very short ultrafast lasers on different applications. And this is the main goal.
So, but they're made here in Porto, because I understand your company is here in Porto?
Yes, the part of the laser, we are doing it here in Porto, also the diagnostics, but the part of the applications, part is being done at INL, part is being done at Adela's lab, and also in Andrew's lab.
So we have some partners here. Are any other partners testing things out in the US other than the UT Austin, Andrew and Adela? Is anyone else testing this?
At the moment, no, because this is a project exactly to be able to validate the concept and see if we can extend this to other applications. So the idea is afterwards, yes, we can extend even in Portugal, that are a lot of microscopy laboratories, to be able to test this new technology on their labs.
And when are these tests happening? Is it soon? Are they happening now?
Are they happening now?
We don't have the lasers yet, but what we're doing in anticipation of the lasers is basically adapting the microscopes that we've built to allow us to take advantage of the unique capabilities of these lasers. as soon as they arrive here in Austin.
Is that due to happen, I guess, 2022, like mid 2022, or when are you expecting them to arrive?
Yes, at the moment, the INL is testing here in Portugal. And once they finish doing the testing, the systems will travel to the US. And this is expected to happen mid next year.
Oh, okay. Now, this is just a curiosity of mine, but I don't know. I know how to send a package to the US, but I'm wondering, how do you send a laser? Are you using Amazon to do? How does that work?
You can use a courier. The thing is of shipping things to the US, because they are not a sale, is they have to travel in a different way. So we normally, what we do is we use a carneata, so it's like a passport for the system, so they can travel to the US and then they can come back. So this is not a sale, and I mean, there is no point to pay taxes of equipment like this. So this is the way you do it when you have borders.
Now, I understand this project, we are combining lots of knowledge areas here. We're talking about physics, we're talking about life sciences and technology transfer, because that's one of the main goals of this podcast, is actually to talk about the bridge between the companies and academia. So I'd like to ask you, and maybe I'd start with Andrew, what are the main hurdles of working with all of these different fields?
Well, so there are a number of hurdles, but as far as the interdisciplinary work goes, that's something that we're already accustomed to working with. So I'm in the biomedical engineering department at UT Austin, and so we inherently kind of span physics, in my case, neuroscience, others like Adela works in physics, engineering, biology. So we're used to crossing these disciplinary boundaries. Additional, I would say, challenges and opportunities, though, include combining academic research with industry, and also collaborating between the US and Europe and Portugal.
What about your experience, Rosa? What are the main hurdles you, as a person who's representing a company here? Even though I understand you have a background in academia, what are the main hurdles you are experiencing now within this particular project?
I think the most difficult thing to work with academia, pure academia, is the amount of paperwork they have to do to be able to purchase a mirror. I think that is the most painful thing, because Rosa, I cannot do it because the paperwork has to go here and here and there and so on. It's like, oh my God, this is a mirror, come on. I think that is very painful, I would say.
Moment of honesty.
Other than that, I think is very interesting, it's very motivating, because I mean, I think it's good to have the academia and also the company, because the academia is this freestanding, innovation thinking, okay, let's do this, and this and that. And the student says, no, we cannot do everything. We have to do this, and this. So I think that is very interesting, actually.
So actually, I was just about to ask you what the most exciting bit is, but I do think we've got the answers for that. And it's quite lovely to see that we think in different ways when we're talking about academia and industry, but there are convergence points. And I think it's quite interesting to see how creativity flies high in academia, but companies need to get their reality check so we don't fly too high. I think, to wrap up the podcast, usually I ask my guests if they'd like to add anything else that we haven't spoken of, something that they think is relevant to mention. Do you want to add anything, whether about the project or maybe about inspiration to other researchers who are listening to this podcast and who want to have more of these collaborations? Do you have anything to add, or do you think we're good?
I think it's very, very interesting that we have the opportunity to work with two different groups researchers in the US, because even we are very, let's say, we are very familiar working in Europe. It's easier for us to go, travel, have a meeting, etc. But I think working with the US, I think is a very interesting experience, at least for myself, because we never had a project like this. So a transactional project with 5 partners. So I think it's quite interesting and challenging. So I'm really very excited because of that, because you have different cultures, different institutions, different ways of doing things, and then we have to come up all together and make all the goals of the project. So I think it's very interesting that.
Indeed, the collaboration that comes with different cultures and how to get that all together. Andrew, would you like to add anything?
I was, in fact, I was going to add something very similar to what Rosa just said, and that this program is really been unique for us and really beneficial because it has provided this opportunity, one, to work with industry, but in the past, we have worked with a few companies in industry. However, this is the first time we've had the opportunity to work with an international company, which has been very enlightening and fun for us. And then to do all of that during the pandemic has been even more challenging. But we have managed to make progress.
Yeah, you're sending lasers to each other.
So.
You're thriving, so you're doing good. I understand it may have been a challenge to actually adapt to the pandemic, and it has been a challenge to all of us. Thank you so much. I'll end the meeting now.
Thank you.
You're welcome, and have a lovely holiday season.
Merry Christmas and Happy New Year.
Thank you all for listening to this episode of Crosstalks. If you have enjoyed it, and even if you haven't, we would love to hear from you. So make sure to subscribe and leave a review. For additional content, follow us at UT Portugal on Twitter or visit utaustinportugal.org. The ExtreMED project brings together scientists and business people, in particular staff and researchers from Sphere Ultrafast Photonics, the International Iberian Nanotechnology Laboratory, the University of Porto and University of Texas at Austin, Cockrell School of Engineering, Walker Department of Mechanical Engineering, and the College of Natural Sciences, Department of Biomedical Engineering. This project is co-funded by the European Regional Development Fund through Portugal 2020 and the Regional Operational Program Norte 2020, and the Portuguese Foundation for Science and Technology under UT Austin Portugal. The total eligible investment is over 2 million euros, with a public co-funding of approximately 1.95 million euros.